This is a production of Cornell University.
So thanks for Sushan for the invitation. It's my great pleasure to present our recent work on watermelon genomics. Actually, when Sushan asked me to give a talk, I thought I just give a talk at plant biology. Then I checked my slides. It's 3 years ago. So it's sad if I can't provide any updates after 3 years. So here I am. I will prep mostly the work during the past 3 years on watermelon genomics. So here is the outline of my talk today. First, I will provide a background of watermelon and the genus, Citrullus genus. Then I will present our last 5 years effort on reference genome sequencing of watermelon. Then I'll talk about watermelon origin, domestication, and improvement. And then last, I will talk about the pan-genome of watermelon. So as you know, watermelon is a worldwide grown crop. It's a very popular fruit crop. The annual production is about 100 tons. So that's ranked the third among the fruit crops is if you count tomato as a fruit crop, not a vegetable crop. So it's only after tomato and bananas. So watermelon is also a nutritious crop. It contains high levels of lycopene. It's higher than any other fresh fruit and vegetable, even higher than tomato. And the citrulline, this is the compound that originally found from watermelon. That's why it's called citrulline, because the Latin name of Citrullus. So this is an amino acid that is beneficial for health and exercise performance. And also, because of this compound, some media claims watermelon is a natural Viagra. But later we found out that if you want to have the Viagra effect, you have to eat maybe tons of watermelon once. That's not possible, right? But anyway, it does provide health and health benefits for humans. So watermelon belongs to the Cucurbitaceae family. So this family contains about 95 genera and about 1,000 species. So including some well-known fruit crops, fruit and vegetable crops, like cucumber, melon, squash, zucchini, border gourd, all kinds of gourds, right? The watermelon belongs to the Citrullus genus. So this genus contains 7 extant species. The cultivated watermelon belongs to the subspecies vulgaris of the Lanata species. So the Lanata species also contains another subspecies called cultivars. This is a wild form watermelon. So I will spend quite a lot of today's talk on this species. And we also have the 6 wild species, and among them, 3 of them, Myxospermos, Amaras, and Collisensis, has already been widely used in watermelon breeding program to improve the disease resistance and abiotic stress tolerance. The cultivar watermelon has a very narrow genetic diversity because of the bottleneck, domestication bottleneck, and is susceptible to all kinds of diseases and pest infections. So we need novel modern genomic tools to facilitate watermelon research and breeding. So next, I will talk about our effort in reference genome sequencing. I think everyone knows reference genome provides a foundation for breeding, foundational tools for breeding and research. We initiated the watermelon reference genomes project in 2008, led by a group of international scientists, mainly by Yongxu and me. So at that time, we used the short-read technology, Illumina technology, to assemble a high-quality reference genome of watermelon cultivar 1713. So 1713, and 9713 is a cultivar name. It's an East Asia type. It's a highly inbred line. And we published this in 2013. Then with the technology advancement, we generate an improved version of 9713 genome using the long reads and also the banana maps and the high-C maps. this version of the genome was much improved than the previous version. So meantime, in collaboration with Amnon Levy, we generate a high quality genome for Charleston Gray. This is a principal American watermelon cultivar. You can see the difference between 9713 and Charleston Gray. So during the past couple of years, we spent a lot of effort on generating reference genome for wild watermelons. So this is one in collaboration with Susan Renner. We produced a high quality genome of the cultivars, the wild form of watermelon, and which could be the direct progenitor of cultivated watermelon. I will talk about this later. And we also generated the reference genome for the 3 wild species that are used in the breeding program: Nucleospermos, Amaras, and Collisensis. So all these 3 genomes has not been published, but is available in the database we developed. So with this reference genome, they not only provide a valuable resource for fundamental research and breeding, but it also give us the opportunity to understand or study the evolution of watermelon genomes. So by comparative genomic analysis, we can infer the phylogeny of this watermelon species and also the divergence time of this species. And through the comparative genomic analysis, we can also identify large genome rearrangements between genomes of this species. Here shows that there's a interchromosome rearrangement between chromosome 1 and chromosome 4 in Collisensis compared to other 3 species. Okay, with this reference genome, it gives us the opportunity to understand watermelon origin. So how many of you know that which continent that a watermelon was originated?
Africa.
Okay, right. A lot of people thought it's from Asia.
I just thought that last
Because the cucumber and the melon, they originate from Asia. So people also think, oh, watermelon probably also originated from Asia, but not. It's originated from Africa, then distributed to the world. So during domestication and the breeding wild watermelon, you can see here, the wild watermelon is small, pale color, and hard, and bitter. Then become the crisp, large, crisp, sweet, and the red flesh. And the seed and the paintings of watermelon had been found in Egyptian pharaohs' tombs. So this tomb was built like about 4,000 years ago. So this indicates that watermelon has been domesticated for at least 4,000 years. But several critical questions related to watermelon origin and domestication still remain unanswered. The first one is, what is the direct progenitor of cultivated watermelon? So this is a very important question. And the second one is kind of related to the first one. So where in Africa was the watermelon first domesticated? Which part? If you can identify the direct progenitor, then you can also answer the second question. So the first 2 questions can be answered if you can identify the direct progenitor. And also, what are the genetic basis for watermelon domestication? So during the past several years, we spent a lot of effort using the population genomics approach try to address these questions. As I mentioned, identifying the crop wild progenitor is very important. They can help you to understand domestication and also can help you breeding. But identify wild progenitor is quite difficult. The main reason is that the wild progenitor may be distinct. It not exist anymore. And also hybridization with cultivated species and indistinguishable from the feral forms. So there are 4 hypotheses of cultivated watermelon origin. The first one, this is actually was the dominant one since from 1930 to 2013. It's from the Southern Africa citron melon. It's the Amaras. It's this one, the pink one, from Southern Africa. And later we found out this is a mistake. Then the second hypothesis is the Northern African consensus, this area. The third one is West African Igushi melon, Mucospermus, this one. And then the fourth one is the Northeastern Africa, the cultivars from the Sudan region. So as I just showed you that based on the genome sequencing, reference genome analysis, we found this cultivars, the wild form cultivars, are closely related to the cultivated watermelon. This gives an indication that this one could be the direct progenitor of watermelon. Then at the meantime, we did the genome resequencing to sequence the larger populations. So in this study, we report a total of more than 400 accessions that have been deep sequenced. And in another study, we investigate the germ plasmid collection in the USDA National Plant Genome Germ Plasmid System that includes about 1,300 accessions. But this one we use the genotyping by sequencing technology for genotyping. And using the SNPs derived from GBS data for the germ plasmid collection, and also derived from genome resequencing for the more than 400 accessions, we inferred the phylogenetic relationship between. Okay. So we inferred the phylogenetic relationship among all these accessions. One interesting finding is that all the several very few cultivar accessions that we can have at that time. So 4 of them. That's all 4 that we have in the germ plasmid system, and these 2 we included in this study. Actually, these 2 and these 4 are overlap. So in total, we have 4 accessions in the germ plasmid system, and all 4 they clustered at the base of cultivated clay, cultivated watermelon clay. And this gives you a hint that this cultivars could be the direct progenitor, right? Because all of them are closely related to the cultivar species. And also we did, in that study, we did the gene flow analysis. We found there's no gene flow between the amaras, so this one, the citron melon, the amaras to the cultivar species. So this rejects this first hypothesis. And also based on phylogenetic tree, you can see amaras is quite distant from the cultivar species. So later we found out that this was a taxonomy error by a student in some lab. I mean, in Germany, in some lab, they mistakenly labeled cultivar species as amaras. So when they do some other analysis like phenotype, the morphology, and using some SSR markers, they found that one was very close to the cultivar species. So they claim that this amaras was direct progenitor. And that's starting from 1930. Then in 2013, when we published the first reference genome paper, we found out this is an error. There's no way that amaras is the direct progenitor. And also, from this phylogenetic analysis, we can easily reject consensus because the consensus is even further away than amaras compared to the wild cultivar species. So now we left with Igushi melon and the cultivars melon. We are trying to provide more evidence. Okay, so we are trying to provide more evidence to support that cultivar melon was a direct progenitor. Fortunately, Susan Reno got a bag of seeds that are collected from Sudan. It's not easy to collect stuff from Sudan because of war and the traveling condition is very bad. But we fortunately got one bag of seeds, and they sent me a bag of seeds, and also sent another bag to the same seeds to Cecilia at the University of Georgia. So Cecilia lab has a facility to make inbred, to cross the self-cross the seed to pure the material. So then we got 15 seeds from Cecilia, and we grow them at the BTI greenhouse. And we did genome resequencing for all these 15 accessions. And you can see we did the phylogenic analysis, and all 15 was clustered in a separate clade, but it is the most closely related to the domesticated watermelon. This provides another evidence to support that cultivars is a direct progenitor. But and also we detect gene flow between cultivars to the cultivar watermelon. And another hypothesis is that this cultivar melon, they are in wild form, but they could be feral. They domesticated before, but escaped from domestication, become wild. And if that's the case, they should have the similar nucleotide diversity with the cultivar watermelon. We performed the nucleotide diversity analysis, found out that the cultivar melon cultivars has much higher nucleotide diversity than cultivar watermelon. So this indicates that cultivar melon is not like a feral form. It is a direct progenitor of cultivar watermelon. So then we answered the first question, right? Then we know that the cultivar melon was from Sudan, the northeastern area of Africa. Then we can conclude that a watermelon could be first domesticated in Northeast Africa. Okay, with the direct progenitor, then we can answer the question. We can investigate the genetic basis or genomic basis of watermelon domestication and improvement. So, give you a little bit of background. I think most of you already know that during crop domestication, there's a lot of morphological changes or trait changes. So this table lists the main trait changes. The first one, the edible part, like fruit, grain, become larger. Like in tomato, the fruit become almost 100 times larger than their direct progenitor. And the fruit has a high edibility, like the wild watermelon, like cucumber, they're bitter, right? And even wild tomato, they have some bad taste, so you don't want to eat them, right? So now with the modern watermelon, they are non-bitter and also sweet, right? It's more edible. And then they have high levels of seed germination and the growth habit. But one good trait they lost is the tolerance to biotic and abiotic stresses, kind of like shown in this picture. So this is a wild tomato growing in these harsh conditions. There's no way that the cultivated tomato can survive or grow well in this kind of conditions. And these wild tomatoes, they can get fruit, has no problem. So that's why the current breeding program, they want to use wild species to increase the tolerance. So I've also given you several examples on how much the domestication changed the phenotype. So do you guys know what is this crop, the wild crop? Anyone know what is this? Okay, it's a carrot. You can see the difference after domestication. So this one, no, it's eggplants. The wild eggplants. Then this is the early domestication of eggplants. So you can see that's why they call it eggplants. It's like egg, right? So this is a modern eggplant, after domestication and the modern breeding. And this one should be easy to guess, right? Banana. And then the teosinte and May, you can see that during the domestication history, the changes of corn, right? This is the wild one. Then this is a corn found in like 7,000 years ago, then compared to modern one. So we are lucky that we have all kinds of those fruit and crops to eat. Right? Like 7,000 years ago, you have to eat this kind of stuff. Okay, during watermelon domestication and breeding, watermelon breeding can can be classified in 2 stages. The first one is domestication. And this is not a real representation of the landrace. It's a painting from 17th century. That means people in 17th century could eat watermelon like this. Then after improvement to modern sweet dessert watermelon, you can see all kinds of these traits were changed, like sugar content, flesh color, bitterness, texture, aroma, size, everything. So then we have this population genomic sequencing data. We have the reference genome. Then we can use this data to understand the genomic basis of watermelon domestication and breeding. So in this paper, in our 2019 paper, at that time, we don't know which one is the direct progenitor of the cultivated watermelon. And also we don't have much accession of the cultivars to work with. So in this paper, we just use this myxospermos as a representative of the progenitor of Lanatas because its fruit was not domesticated. So this wild species has some kind of domestication for seed consumption, but it's not a real direct progenitor. So there could be some information missing if you use this as a representative of direct progenitor. But at that time, we have no choice. But it still gives you some interesting findings, but you will miss or maybe give you some wrong conclusions, right, using this. So as I mentioned, we got 15 cultivar accessions from Sicilia. We grow them in BTI. And actually my student and postdoc tasted all 15 of them. I also tasted some of them. And they taste like watermelon, non-bitter. None of them are bitter. And they are not sweet. So you can see the Brix, but content is the sugar content is very, very low. The Brix is 0.2 to 3.2. The modern cultivar is like 10 or 11. It's much higher than the cultivars. And we also saw some pink-colored, pink-flesh watermelon in cultivars. So, and this provide us the this accessions and the genome sequences provide us to opportunity to revisit watermelon domestication. So we compared the cultivar melon with the cultivar watermelon with landrace, then landrace with modern cultivar to identify domestication sweeps and the improvement sweeps. And we identified about 20 million base pairs of the genome regions that are under selection, about 400 and 700 genes that are in this selective region, or these genes are under selection. So as you know, in watermelon, fruit sweetness is the most interesting target or trace for domestication. And even now for breeding, people still talk a lot about the sweetness. And this slide shows the sugar content in different species. And in landrace and modern cultivar, the sugar content is much higher than the cultivars and other wild forms. So we identified a genome region that under selection that overlap with the known sugar QTLs, like this QTLs, the QTLs in blue, was under selection during both domestication and improvement. And this QTL was under selection during domestication, but not in improvement. This one was in improvement, but not in domestication. So fruit bitterness. So we found out that all cultivars are non-bitter. And also the one major gene regulating fruit bitterness in watermelon, that's the LBT gene. You can see the nucleotide diversity around this gene has no difference between the cultivars and cultivated watermelon. But the nuclear diversity was much higher in the Mycosophomus. So that means this gene was not selected during domestication. This kind of like against previous studies or previous thoughts, because people always thought in cucurbits, bitterness is a major selection domestication trait. But our study says it's not, at least in watermelon. It's not a domestication trait because they already being fixed in the wild progenitor. And how they become fixed in this wild progenitor, that's an interesting question. And so far we don't know. We don't know why this trait was fixed in the wild form. And then flesh color, we found out that the wild progenitor already have the red-fleshed flesh color. This is also different from previous studies because people thought about the flesh color was introduced during domestication. Because all the other wild forms, except the cultivars, they don't have red flesh, they are white flesh. So previous study using these materials, they thought that domestication, the red color was happened during domestication or early stage of domestication. But actually we found out it's not the case. The red flesh color already in the wild progenitor of cultivated watermelon. And this one gene, the LCBY, lectin beta cyclase, this is a major gene controlling watermelon fruit flesh color. And you can see the diversity of this surrounding this gene was decreased when you compare the cultivar melon. This brown one is cultivar melon, and then landrace is cultivated one, and also the modern cultivars. So this indicates that this gene was under selection. And also the red color was a selection trait, domestication trait. So combine all this evidence, we think that watermelon domestication could have started with red fleshed, non-bitter, and non-sweet. Right? But this non-sweet, and also this variation of the Brax or sugar soluble solid content of the in the wild progenitor. So they may start with the red one because also the red one has higher Brax values. They may start this for domestication. Okay, meantime, as I mentioned before, the wild watermelons had been used in modern breeding program to increase the abiotic and abiotic stress tolerance. So using this genome sequencing data, we are trying to identify genome regions, the footprint of integration in the modern watermelon genome. So we did find a region on chromosome 6 that was integrated from the Amaras, the citron melon. And this region contains genes related to powdery mildew resistance or tolerance to hypoxia or low phosphate conditions. This is as expected because the reason we bring this in is to increase this tolerance. And we also identify a region on chromosome 4 that will integrate from coalescences. And this one was contains the QTL that are related to fung resistance. Okay, so next I will talk about the pan-genomes of cultivated and whited watermelons. So you can see from this picture, there's a wide phenotypic variation of even within cultivated watermelon. So this indicates that one genome couldn't provide a comprehensive reference to evaluate the genetic diversity of watermelon. So that's why we need a pan-genome. So as I mentioned before, we have reference genome for 9713, Charleston Gray, cultivars, and these 3 wild species. And we also reported the genome resequencing at 414 accessions in 2019. And during the past several years, a couple of years, we generate quite a lot of new genome resequencing, mainly on the wild species. And you can see here the genome resequencing was mainly on cultivar. So we are focusing on generating genome resequencing on wild species. So here is the final resequencing data, the summary of final resequencing data. We have more than 400, we have close to 550 accessions that have been deep sequenced in watermelon. And the reference genome and the genome resequencing data provide a solid foundation for our pan-genome work. So we use the map to pan approach to construct the watermelon pan-genome. And we de novo assembly for each accession, we de novo assembly, we generate the contiguous map to reference. Then we identify novel sequences that are not in the reference genome and novel genes. And because the high divergence between these different species, so we first construct a pan-genome for each species because we have reference genome for each species. So in Lanatas, we have identified about 2,288 novel genes. Myxosomos, we identified much less, about 600 genes, and 2,000 novel genes in Amaras, and 2,500 genes in collisensis. And the genes, we checked the presence and absence variation of each gene in the population, and we found out that the core genes in different Lanatas Ceratolus species are quite high. You can see in Lanatas 85%. The core genes that means the genes present in all accessions in that species. Myxosomos 97, this one is very high, and Amaras 90%, and collisensis 88%. So after we construct these 4 species-specific pan-genomes, we integrate them into a super pan-genome, watermelon super pan-genome. So what we do, we lift over genes among the 4 species to try to identify the orthological relationships between each pan-genome or among those pan-genomes. We also use synthetic gene blocks to help to identify or to establish orthological relationships. So in the end, we identified about 38,000 orthological gene groups in the pan-genome. And most of them, they have the synthetic orthological relationships. And some of them, they are specific to one species. So the pan-genome has much less core gene content. It's 64%. So this indicates the diverse genetic makeup among the 4 species. This is understandable because interspecies variation is much bigger than within species variations. And we also found that during domestication, this is a cultivar melon, this is a landrace and a cultivar, we found there's a trend of gene loss during domestication. This also found in tomato, the pan-genome we reported in 2019. So we checked the gene, we analyzed the gene presence, absence, variation pattern, and identified genes has a highly differentiated frequency, highly different frequency among different populations. And we did not find much genes that are showing highly different allele or gene presence, absence, variation frequency during domestication and during improvement. So this is also understandable because the history is very short. Like domestication is like several thousand years, and the improvement just several hundred years compared to this species divergence that's million years ago, several million years ago. But we analyze this data set, the present absence variation, we did find quite a lot of interesting stuff. So here I will give you a couple of examples. The first one is a disease-resistant gene. We find quite a lot of genes, these disease-resistant genes, that are missing in cultivar and also in their direct progenitor, or very low frequency. So this one means that this gene present in all accessions we investigate in colosynthesis species. So this like this 0.937, that means 93% of accessions in this species contain this gene. So there means that no accession. The gene is absent in all accessions. You can see we have a huge amount of accessions in cultivar, 243, 88 in landrace, and all of them does not contain these genes. And this provide a valuable resource for target breeding, for disease resistance, right? We know that these are known target disease resistant genes, and they are missing in modern cultivar, and then we can introduce them to the modern cultivar to improve the disease resistance. So the other one is a TH2 gene. So this gene is a tonoplast sugar transporter. And based on our analysis, we found this gene is a tandem duplication in cultivar species, at least in 9713 genome. And then we checked this tandem duplication in different watermelon populations, and found out that in wild species, almost all of them are single copy. It's not a tandem duplication, and except one of them. All these populations, only one of the accessions have the tandem duplication. But you can see in landrace, the tandem duplication has become much more common. And in cultivar, it's almost fixed. Only very few has single copies. And we identified one codofinous accession has 2 copies. And it turns out that this accession has the highest sugar content among all these 15 codofinous accessions. And we also checked in cultivar and the landrace, we checked the sugar content in accessions with tandem duplication, and in accessions contain one single copy. We found out that the tandem duplication has much higher sugar content than the accessions with one single copy. And we checked this gene, the nuclear diversity of this gene, and found out that the nuclear diversity of this gene has dramatically reduced during domestication. That means this gene is a domestication target. Okay, the pan-genome we constructed was based on map-to-pan approach. And the next couple of slides, I will briefly mention our ongoing work using the graph pan-genome. So the graph pan-genome can they can put the insertions, deletions into one graph. The map-to-pan approach is a linear representation of the genome. So this is a graph pan-genome, a graph representation of multiple genomes. And it has a lot of advantages, and like single coordinate system, it can improve read mapping and variant detection, but it requires multiple reference grid genomes. So this, like a couple years ago, this is not realistic because of the technical limitation. But now it's become feasible, like the HiFi, due to the HiFi technology. It can generate the read length of 15 KB. That's 15 times of Sanger sequencing, about 150 times of Illumina sequencing. And the base accuracy is 99.5%. It's equivalent to Illumina sequencing and Sanger sequencing. So this gives us a lot of opportunity to generate multiple reference genomes. So in collaboration with Dr. Yongxu, we already generate more than 130 reference genomes for watermelon. It's all reference level using HiFi. So this is the number shows the number of accessions that are used for reference genome sequencing in each species. So we only have one in Remii, 1 or 2 in Remii, Roses, and Nordiana. So because we in the germplasm system, we only have this accession available. So that's why we only have 1 or 2 reference genome for this species. And this slide shows, this table shows that the number of contiguous that are assembled in each chromosome. And you can see in most chromosomes, those in green, the entire chromosome was assembled in one contigue. The biggest one could be 4, right? So that's still, oh, this one, there's 1 6. But in most of the chromosomes were assembled in 1 or 2 contiguous, thanks to the HiFi technology. And my student Hong Ho recently identified structural variations of using this reference genome, the whole genome alignments, using the 9713 genome as a reference. We identified quite a few insertions and deletions, and it's surprisingly that we got a huge amount of deletions and insertions in wild species. Almost 1/3 of the genome, the size, has insertion and deletions. So we are in the process of constructing a graph-based pan-genome by integrating this reference genome and the structure variations into one single graph. And this one single graph can be used to genotype the resequencing data we generate, like this 547. And also we are in the process of generating the genome resequencing of a core collection we have developed. And I'm pretty sure that this comprehensive data will give you a much more broad or in-depth knowledge of watermelon domestication and the improvement related to these traits. So with that, I would like to thank people in my lab, the wonderful graduate students and the postdocs, and also my collaborators, and the funding from USDA QCAP project and the National Science Foundation. Thanks for your attention. I would like to take any questions you have. Yes. Yeah.
Do you think the reduction of bitterness in the progenitor and the increase in the color red is due to selection from mammals that might have distributed the seeds?
So you mean the non-bitterness, right? The reduction of bitterness and the increase of color could be caused by the animal distribution of the seeds. Yeah, it's possible, but I'm thinking why not happen in other wild species? Only in this wild species.
They grow together. They overlap their distributions.
Like colosynthesis overlap with the cultivars. But it could be a reason, but we don't have much evidence. Yeah. It's possible. Yeah.
Hi, very impressive talk. Yeah. So I was wondering, so if the modern watermelons has been domesticated in Africa before they just distributed into Asia or Europe or America, or they just domesticated in Asia or Europe or America.
Okay.
Yeah. This is the first question. Another question is like more general. So I was wondering what kind of principle or general principle to decide or to determine the origin of the modern cultivar of the watermelon. So because it's based on the close, it's based on the wild watermelon with the closest genome with the modern watermelon, or just some other principle?
Okay.
Yeah.
So the first question is whether the watermelon was domesticated first in Africa, then distributed to the world, then just wild distributed to the world, then domesticated in in Asia.
Locally or just?
Yeah. I think the answer is that the first domesticated in Africa.
Okay.
Then the early domesticates were brought to the world. Then each continent, like Asia, America, they have their own breeding program to improve watermelon. Right? And there's no way that the wild species bring the wild species to the world than domestication. Yeah, this should be clear.
Yeah.
So the second question, can you repeat your question again?
Yeah, basically, so this is a general question. So like, what is
How-
the general principle to determine
The origin.
like origin of one of the cultivar or origin of watermelon? So
Yeah, I mean, to determine the crop origin is quite difficult, as I mentioned. Mainly people using the existing wild species to analyze their morphological difference, their genetic difference. If they find a wild species that are most closely related to the cultivar species, and one possibility is that this wild species could be feral. It could be domesticated, then escaped from domestication, become wild. And then you have to rule out this possibility. And then people, based on this analysis, they claim that this one could be, I mean, it's not 100%, could be the direct progenitor of cultivated species. Because why you can't 100% sure? Because there could be another species, wild species, that are not existing anymore.
yeah.
They extincted, then you can't identify them. But if you find, like in our case, if you find a wild form that are highly close related to the cultivar species, then using that, even it's not 100% as a wild progenitor, using this as a representative of wild progenitor for analysis, it can still give you a lot of information on domestication. Yeah.
That was a great talk. So my question is kind of about, generally speaking, not just about watermelon, about plant breeding and selecting the right traits for domestication. So is there a sort of trade-off between pest resistance and drought resistance and some of the things you want to breed back into watermelon and all the other agriculturally sweetness, the other things we really like about the crop? Is there a general trade-off there, and that's an issue for all crop development?
Yeah, very good question. So actually, from genetic point of view, there could be a linkage drag. Yeah. So there could be tightly linked of two traits, right? I have a fruit size trait that is tightly linked with a susceptible trait, right? I want to increase fruit size. Then at the meantime, you bring the susceptible allele to the modern cultivar. So that's quite a lot. So there are some studies in tomato, they found quite a lot of linkage between the two traits. So another one is that for some traits, people don't like, right? But they can resistant to pathogens or insects, like beetle.
Yeah, right.
Bitterness,
Yeah,
right?
good sales.
And then you fruit is bitter, then animal or pets, they don't want to eat. But people also don't want it. They get rid of the bitterness, then they decrease the resistance to pests.
Got it. Okay.
Okay, yeah, thank you very much. Actually, this is a really great seminar, and let's give applause to the
Thank you. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.